The enzyme we chose is aldehyde dehydrogenase. I'm Ksenia.
And I'm Brooke. The discovery of aldehyde dehydrogenase cannot be linked to any individual, and it was identified through studies on alcohol and aldehyde metabolism in the mid-20th century. Aldehyde dehydrogenase plays a vital role in detoxifying toxic aldehydes such as acetaldehyde, and advances in enzymology has later revealed multiple isoforms and their links to these diseases.
The structure of aldehyde dehydrogenase, or ALDH, has a main chain residue of 501, and the enzyme has a mass of 54,862 Da, which is approximately 54 Da, and is a key enzyme in aldehyde detoxification pathways. It typically functions as dimers or tetramers, depending on the isoform.
So in order to break down ALDH a bit, it's a polyformic enzyme that oxidizes aldehydes into carboxylic acids, which can lead to an increased function in biological processes. It is found in three types of classes in the cytosol, mitochondria, and some microsomes, depending on the mammal. It is a member of the superfamily of NADP plus dependent oxoreductases and is a crucial enzyme that protects the heart from oxidative stress as well as alcohol in the body. And as well, ALDH aids in the formation of essential molecules such as retinic acid.
Aldehyde dehydrogenase deficiencies can lead to various health issues due to the inability to properly metabolize aldehydes, particularly in processes such as alcohol detoxification and cellular detoxification. The consequences of ALDH deficiencies include accumulation of toxic aldehydes, alcohol sensitive and intolerances, congenital ALDH deficiencies disorders, an increased risk of alcohol-related diseases, and impaired cellular detoxification. The key point is that the different isomers of ALDH have distinct roles in different tissues, so deficiencies can manifest differently depending on the specific enzyme involved.
A conserved domain is a recurring unit in a polypeptide chain that is made up of a conserved sequin pattern or motif. That being said, these enzymes exist and function as homodimers and homotetramers that consist of three main conserved domains. These serve their overall function as well as classifying them within a superfamily. These consist of the catalytic domain, which is responsible for the enzyme's catalytic activity. It facilitates the conversion of substrates into products, typically involving redox reactions. The NADP binding domain, which binds NAD plus or NADP plus cofactors essential for electron transfer during the enzymatic process. And the final one is the oligomerization domain. This facilitates the assembly of the enzyme into its functional multimeric form, whether it's the dimer or the tetradimer, ensuring stability and efficient activities.
Functions of ALDH are the detoxification, where ALDH plays a role in detoxifying harmful aldehydes, such as acetaldehyde, which is produced during alcohol metabolism or oxidative stress. By converting aldehydes into non-toxic carboxylic acids, this reduces cellular damage and maintains cellular health. The next is the stem cell regulation, where ALDH is essential for maintaining stem cell health and function by regulating the balance between stem cell self-renewal and differentiation. It actively supports stem cell longevity, enabling tissue regeneration and repair in various organs. Another is the biosynthesis, where ALDH contributes to biosynthesis by managing aldehyde intermediates involved in the production of essential cellular components like lipids, proteins, and nucleic acids. Its role ensures the proper function of metabolic pathways necessary for cell growth and energy production. The last is cardiovascular protection, where ALDH protects the cardiovascular system by reducing oxidative stress, enhancing mitochondrial function, and regulating autophagy. It has been shown to protect against diabetic cardiomyopathy by maintaining heart cell health and function.
So moving on to a little bit more about the mechanism. The mechanism of aldehyde dehydrogenase involves 5 key catalytic steps within the enzyme's active site. So the first being the thiol activation, where the catalytic thiol group, or the cysteine 302, is activated through water-mediated deprotonation, which is facilitated by glucose 268. Following that is the nucleophilic attack. The activated thiol attacks the aldehyde, forming a tetrahedral thiomecetal intermediate. Next is the hydride transfer. Deacylation occurs coupled with the transfer of the hydride to the pyridine group of the NADP, which produces the thioester intermediate. Next is the thioester hydrolysis, which is when the thioester is hydrolyzed, aided by an ordered water molecule. And finally is the regeneration step. The enzyme regenerates as NADPH dissociates, which allows new NADP binding. A water molecule plays a role in both the thiol activation and the thioester hydrolysis, ensuring efficient catalysis occurs.
As seen below, the sequence of aldehyde dehydrogenase is fairly balanced between polar and hydrophobic residues, which is crucial for maintaining both enzyme structure, integrity, and catalytic efficiency. The hydrophobic residues help stabilize the enzyme's three-dimensional structure by forming a stable core, and the polar residues facilitate interactions with substrates, cofactors, and water molecules, enabling efficient catalysis and proper enzyme function. This balance ensures that aldehyde dehydrogenase can effectively carry out its role in the detoxification of aldehydes and other metabolic processes.
Go into a little bit more depth on the catalytic activity. The catalytic activity of aldehyde dehydrogenase is the oxidation of an aldehyde to a carboxylate, which was previously stated before. This reaction requires NAD+ as a cofactor and involves the consumption of water. The products are a carboxylate ion and protons and the reduced form of the cofactor NADH. This enzymatic reaction is essential in detoxification and metabolism, converting toxic aldehydes into less reactive and more easily excitable carboxylic acids.
In an article, a novel protective mechanism for mitochondrial aldehyde dehydrogenase, specifically the ALDH2 in type 1 diabetes induced cardiac dysfunction, a role of AMPK regenerated autophagy, with the purpose of investigating how ALDH2 protects against diabetes-induced cardiac injury with a focus in mechanisms of autophagy regulation via the AMPK FOXO3A pathway, and a role of ALDH in reducing mitochondrial damage and cardiac dysfunction. And the significance of this is of demonstrating the therapeutic potential of ALDH2 activators by treating diabetic heart disease.
So to talk more about the methods of this study, the study investigated the effects of ALDH2 overexpression in diabetes on cardiomyocyte function as transgenic mice and H9C2 cells. Male FVB and ALDH2 transgenic mice were induced into diabetes using streptozotocin, which is abbreviated by STZ, with cardiomyocytes isolated for functional assessment. Cardiomyocyte mechanics such as a contraction and relaxation were measured using an ion optics system, and an intracellular calcium handling was evaluated using Ferna-2 fluorescence. Histological analysis was performed on paraffin-embedded heart sections, while cell culture experiments with H9C2 cells assessed glucose and ALDH2 activator effects on autophagy, apoptosis, mitochondrial membrane potential, and protein expression via Western blotting. Additional assays included MTT for cell viability, TUNEL for apoptosis, and GFP-LC3 adenovirus transfection to quantity autophagy-related changes. Statistical analysis also employed ANOVA with Tukey's post hoc test, considering P was less than 0.05 as significant.
Results from this article showed that the overexpression of ALDH2 in diabetic mice showed a protective effect against cardiac dysfunction induced by diabetes. This indicates that the ALDH2 may help mitigate mitochondrial damage in the heart, which is crucial for managing diabetic cardiac complications. And the ALDH2 regulates autophagy via the AMPK FOXO3A pathway. This pathway is essential for clearing damaged cellular components, contributing to cardiomyopathy survival under stress. This suggests that ALDH2's role in autophagy could be a potential therapeutic target for diabetic heart disease by improving mitochondrial health.
In addition, the study also observed improved cardiomyocyte contraction and relaxation and better intercellular calcium handling in ALDH2 transgenic mice compared to controls, which shows that ALDH2 overexpression directly supports heart muscle function and offering potential for improving heart health in diabetic patients. The research also suggests that ALDH2 activators could serve as a potential treatment for diabetic heart disease. This opens the door to new therapeutic options aimed at improving heart function in individuals with diabetes.
Key findings from this article is that ALDH2 protects against diabetes-induced cardiac dysfunction. The protective effects are mediated via an AMPK-dependent autophagy, and that ALH2 activation restores cardiac mechanical functions, intercellular Ca2 plus regulation, and prevents autophasis. ALDA1 and ALDH2 activator mimics these effects by enhancing AMPK phosphorylation and modulating FOXO3A and ULK1 signaling. Some therapeutic implications are that ALDH2 activation holds promise for treating diabetic cardiomyopathy and hyperglycemic cardiotoxicity. Further studies are needed to explore clinical applications and molecular mechanisms, but overall, ALHD2's regulation of autophagies offers a potential therapeutic strategy for improving heart function in diabetes. Thank you.